To the last week of classes. Thank you for being here and still alive. Yes, most people, yeah, survived Thanksgiving. Yeah, I need a vacation. Really, I do. Okay. So I've been getting a lot of emails asking what's up with this exam number 5, if it is the same thing as the final, do you need to take exam number 5? So again, let's go over this. Everybody's awake? Good? Yes? And people in the back, can you hear me here? Say hi. Yes, let's go for the action potential, like, guess it's not gonna work. Okay, so let's pay attention here. Today is the 27th, right? Yes. Yes, good. So we have today's lecture. Hopefully you're gonna finish everything about chapter 11 that you need to know. Then we have one more lecture on Wednesday, the 29th. Then that's it for the lectures. You're going to, I thought I had the pointer here, but it's only in my computer. Okay, so here, we are here. Then we have only one more lecture. Then you are going to have a whole week here to study, right? And I thought that I could be lecturing on that week too. No, not really. My fault, sorry. So you'll get to study on that week. Then we have our 5th exam that is not optional. That means you have to take the 5th exam. The good news is that it's not cumulative. So it's the material from after the last exam until whatever we are going to cover there on the 29th. Okay? Now, on this day, you're going to come to this auditorium. You're going to do the same thing that we have done so far with all the other exams, right? You're going to put your book bag under your seat. You take out all these devices in your ears and that you're communicating with people from other planets, I don't know, right? So you put everything under your seat, no watches, no phones. Take your number 5 exam, right? As soon as you're done with that exam, you hand it in. Then you're going to have the option of taking the cumulative exam, right? You hand in number 5, then you say, okay, I want to take the cumulative. You pick up your cumulative exam. Make sure you bring 2 scantrons a day, right? Because you're going to have to label them properly, right? Use them both, okay? So then what you're going to do, you're going to go to your seat. You're going to pick up your cheat sheet, right, tongue twister. Remember, you can make your own cheat sheet, right? Normal size, 8.5 by 11, and you can make your notes for your cumulative exam, not for exam number 5. Everybody clear with that one? Yes. Yeah. So then you're going to have plenty of time during that period because it's a very long time, right? So you go back to your seat, you take the cumulative exam, and then you hand that in, and then you're done. If your grades are so good that you don't feel that you need the cumulative exam, don't take it. You're going to need 5 grades from 5 exams, okay? If you decide to take the cumulative exam, then you can drop your lowest grade. If your cumulative exam is your lowest grade, that's the one you're going to drop, okay? Now, if you have missed an exam, anybody here missed an exam? Well, okay, don't have to put your hand up if you missed the exam. But if you have missed an exam, make sure that I know about it before this week here, okay? Because I've been very lenient allowing people to take makeup exams, okay? And I want to give everybody the same treatment and the same opportunity, the same way they've been very lenient with late homework or late quizzes. Nothing is late anymore, okay? So make sure you get in touch with me. You want to make sure that you give yourself the best chances of getting a good grade in the class, okay? Everybody clear with the exams? All right, so now, friends, all right, what is happening here with this unipolar neuron? You have two axons in this guy, right? So this is not the bipolar, this is the unipolar. There are three types of neurons that you should know, right? It should be second nature of nomenclature in your head. No pun intended, right? So here, you have the unipolar, you have the bipolar, and you have the multipolar. What do you know about the unipolar? You look at the cell body over here, and it's like on the side of these processes, right? You call processes these little branches that come out of the cell body, right? So this guy here has axon this way, has axon this way. The interesting thing here is that the triggering zone for the action potential is actually around here, so the flow of the action potential goes down this way. Okay, so this here is actually is an axon. Okay, so axon here, axon there. When you compare here with a bipolar neuron, so now you have this cell body, remember it's also called the soma, right? The cell body more or less in the middle. And here you also have two processes, but one of them is a dendrite and the other one is the axon. And then the cell body is in. Okay, so this is your bipolar neuron. And then you have here a multipolar neuron that is really the most popular one, right? In which you have the cell body that is receiving information from a lot of dendrites, right? And then this cell body has one axon coming out of it, right? And you're going to find the axon hillock around here. So the information will come from these sides here, and it's going to be transmitted down the axon. Okay. Everybody clear with that? Those types? Piece of cake, right? All right. Okay, so this is a pretty picture. So here, neuron type. So you do have your multipolar here that we just mentioned. Now, where are you going to find these types of neurons, right? So that's actually our next slide. But here, just to make sure that we know what's going on here, this is straight from the book, right? So here you have your dendrites, your cell body, your axon going this way. So here is your dendrite on your bipolar. So dendrite this way, cell body in the middle, and then axon going down that way. Information goes down this way, right? And there you have your unipolar. The unipolar neuron is also called pseudo-unipolar, okay? Now, I'm happy just with unipolar. Okay. About this multipolar neuron and your bipolar neuron. Now, with this unipolar neuron, I'm not really going to emphasize much on it, but one thing to pay attention to these guys here, to these unipolar or pseudo-unipolar neurons, is that the cell body is outside of the CNS. Okay, so these guys here, the unipolar, remember it has a dendrite here and it has the axon, right? I hope that one is right. Two axons. See, I always got confused with that one. So here you have the most important thing here, you have the cell body. The cell body is outside of the central nervous system. Okay. Now, when you compare here an example of your bipolar neuron with special senses like the eye, for example, so this is also in the special sense of the eye, it is considered part of the CNS. Okay, so here the body is inside the CNS, but here the body of that unipolar neuron is outside of the CNS. So now we go to your first iClicker question. So overall function of your nervous system, right? So it has three overlapping functions. Which of the following represents the logical sequence of these three functions? So pay attention here. What is the function, overall function of, in which you have a sensory input that is going to be integrated, and then you're going to have some type of motor output, or yeah. No, that's going to be at the end because the extra credit is going to be, everything is calculated based on points, right? So after your, exam number 5 and final exam, we're going to sum up all the points and then we're going to add whatever points you got for the extra credit. But you should know more or less how many points you have, right? You can see that, right? Everybody can see that? Oh, all right, cool. Okay. Okay, so continuing here. Terms that you need to know about the nervous system, okay? Concerning the cell body, right? So most of the neuron cell bodies are in the CNS, but some of them are not. Okay, so if you have cell bodies, a cluster, a bunch of cell bodies that are located in the CNS, you call that nuclei. Okay, so a nucleus is a cluster of these cell bodies, and a bunch of clusters, those are the nuclei. All right, this is bodies in the CNS. Now, if you have cluster of cell bodies that are in the peripheral nervous system, so you don't call those nuclei, you call those ganglia or ganglii, right? It's the proper term since nuclei is plural. So these are ganglia or ganglii. Different terminology just to tell you where the cell body is. Right. Now, concerning the processes of those neurons, right? So you know what the processes are. They can be dendrites, they can be axons, right? So you're going to have these processes in CNS, in PNS, okay? Now they're going to be called different things. When they are in the CNS, okay, so bundle of processes in the CNS, you call those tracts, okay? When they are in the peripheral nervous system, then you call those nerves, right? So nerves in the periphery, tracts are in the CNS. And this is interesting because I always remember one time that I had a small car accident. It wasn't anything serious. It was one of those things that I wasn't really paying attention. It was one of those times that I was managing, a postdoc position with having 2 kids and having the kids in extracurricular activities. So my kids had swimming class and we were getting late for swimming class and I'm paying attention to that and I'm holding the bottle in the back here for one of the kid and tell the other kid not to be smacking the first kid. So it was kind of a mess. I would not advise anybody to drive in that kind of a mental state. And then what happened? The light is red, there is somebody in front of me and I'm waiting for the light to turn green, like impatiently waiting, right? So if you're making a thought process, if you're deciding on something, those have to do with neurons in your central nervous system. So you should have tell the person, you have the track. Right? That would sound a lot more intellectual than you have the nerve. Right? Anyway, so you got the difference, right? So somebody get an extra credit and write me a test question for that. Thank you. All right. Now, more about the processes. We know about those dendrites, axons. Everybody is happy with them? So we shall move on. Okay. So, in these neuronal processes, okay, especially when you're talking about axon movement, stuff that moves down the axon or up the axon, okay? So what's going to happen here? You have these transporting mechanisms that are very, very important to carry vesicles. This looks big here, but it's tiny, very, very tiny. Remember, vesicles are tiny, vacuoles are big, right? So this guy is a vesicle, all right? This guy is going to transport neurotransmitters, mainly, okay? So remember that the neurotransmitters, well, in this chapter, at the end of the chapter, we have a lot of information on neurotransmitters, but we're not going to get into that. It's a lot of information and it gets very confusing. And if you take a neuroscience class, most likely you're going to see that again. Okay, so I'm going to mention just a few of them that you absolutely have to know. And you can forget about the ones that I don't mention. Okay? Actually, in your homework, you don't even have a lot of neurotransmitter questions, right? Okay, so what happens here? In your neuron, most of your neurotransmitters are going to be synthesized in the body. You gotta know that, right? They are synthesized in the body. Then they get packed in these vesicles and they get transported to where they have to go, which is usually the end of the axon, right? So they get down the axon, this long axon, they get transported. This process is not spontaneous. It does not happen by diffusion, because if you would wait for this thing to happen by diffusion, it would never happen, okay? Because you might have the same number of vesicles that you have in the body than you have, close to the axon terminal, or even worse, you might have more vesicles at the terminal than you have in the body, and then diffusion would happen backwards. So that would not work. So this is active transport, okay? Active transport. Different type than when you talk about active transport through a membrane. That's a different thing, right? But what they have in common, they use energy, mainly ATP. Everybody got that? It uses energy to transport these vesicles. Now, the type of transport that goes from the body down the axon, that is called anterograde process. So that goes away from the cell body and towards the end of that axon. What gets transported? All right, basically everything. You also have a type of transport that's called retrograde transport, which we had mentioned before, right? By the way, this is a little review of your cumulative exam, just in case you have not noticed yet, right? All right, including the part of active transport in the membrane. That's kind of stuff you should know, right? All right, so here, if you have retrograde transport, that means you're going to bring vesicles from the end of the axon back towards the cell body. Now, the type of molecular machinery that is used in this transport, they are different. So you have, for example, here kinesin proteins that walk along microtubules that are going from the cell body that is way up there somewhere towards the axon, and down the axon that is going to be down like this way, okay? So here you have a little molecular machinery, and at the end here of this protein, of this kinesin, you have ATP binding pockets and ATPase activity. So you remember how the muscle was working? Like bind ATP and get a power stroke, and then you have that myosin. Remember myosin? Myosin head? Okay. So the myosin would dephosphorylate the ATP, you get ADP, and then you have the cocking of the head. So you have that movement in the myosin head. So same thing happens here with kinesin, okay? So now that is happening here. And when you have ATP that binds, then you have detachment of that little part from the microtubule, and then this thing hydrolyzes ATP. Then you have the cocking of the foot. In this case, it's not the head, right? Because it kind of looks like a little foot, and then binding again. And this, you see, this kinesin has two chains, and they keep on exchanging when each one is going to be bound to ATP or to ADP. And this is really neat, because you see that the binding and unbinding of the kinesin foot to the microtubule, it gives this walking motion. Okay, and at the other side of this kinesin molecule, you have the attachment of this huge vesicle. Okay, so this tiny little thing that you can compare to a little ant, right, is carrying this huge vesicle down the axon. All right, so what you have to know here, anterograde process, retrograde process. That goes back. So here I have added the video of how kinesin works, in case you haven't seen that before, right? You see the little feet over there? Isn't that neat? So you have this walking motion of the kinesin on the microtubule, and these long microtubules that work like tracks. So you have a little walking thing on the track. And here is another question. Movement of substances towards the cell body of a neuron is termed which one? That is your retrograde movement. Okay, so this is an interesting one with homeostatic or clinical correlation, right? Or homeostatic imbalance, right? So there are certain viruses, for example, polio or rabies, and also herpes simplex virus. This is a very, very interesting one, like herpes simplex 1 or 2, right? And then you have the other types of herpes viruses, like chickenpox, that's herpes 3, right? That those they damage the type of transport, the axonal transport of neurons that they are able to infect. And then you have certain toxins like tetanus toxin that is produced by a bacterium, okay? That can also damage nerve impulses and nerve function. Now, here finds the bacterium that produces tetanus toxin. Do you know? A rust, like metal, some type of metal. If you have rusty nail or something like that, right? So it is important that you have your vaccines up to date. So the vaccine that prevents tetanus is DTaP, that is diphtheria, tetanus, and pertussis. Yeah, pertussis is also called whooping cough. Have you heard of whooping cough? It's really bad. Like if you catch it, some people cough so hard that they break their ribs because of that whooping cough, like really, really bad. So this vaccine is given in a regular vaccination schedule. And the thing is that with tetanus toxin vaccine is a type of vaccine that is not as effective as some other types of vaccines because it's against the toxin itself. There's a whole little trick that is done to develop this vaccine. The bad thing about it is that it wears out with time. So the period of time that it gets to be effective is at the most 5 years. So every 5 years you should get a tetanus booster shot. Okay, just because the antibodies don't stay forever, the memory cells don't remember forever. Okay, so after 5 years or so they're gone. So you have to get a booster shot. Now, it can damage your nerves and you can die, or luckily just lose a limb. All right. Now, there are some research that is using this type of retrograde system. So this is going to damage retrograde axonal transport, right? So here's tetanus toxin I talked about here. Now, in relation to that, there are some research that is being done that is using this type of mechanism to plan and develop some genetic therapeutic drugs. Right, so this is basically on the initial stages, but the same principle that is used by viruses and by bacteria, the same principle now is being used to develop some type of delivery mechanism that can correct for certain types of genetic diseases. Okay. So functional classification of neurons. Okay, you have sensory neurons. Those are going to send impulses from sensory receptors towards the CNS. Okay, so go from the senses into the CNS. So mostly all of those sensory neurons are unipolar, right? Remember the unipolar guy? So the cell bodies here are located in ganglia. Now this is important to remember, okay? Cell bodies are in ganglia, in peripheral nervous system. One easy way that I have to remember that ganglia is part of peripheral nervous system, that I say, well, all the gangs will go like to the periphery, you know, like gangs I'm talking about, you know, gangs go out to the periphery, so ganglia are in the periphery, and those have to do with the cell bodies. All right. Those are your sensory neurons. Then you have motor neurons. Those are going to carry the impulses from the CNS to effector places, right? From CNS to something else, motor neurons. Like you have your motor neuron that goes through your muscles, right? Those mainly are multipolar. Remember what this guy looks like, the typical picture of a neuron. with a lot of dendrites in the cell body and one axon, most of the time myelinated, right? So those cell bodies are inside your central nervous system, except some of them. There are always exceptions, right? So here you say multipolar, most of those are in the CNS. And then you have other types of neurons that are interneurons. Those have another name as well. It's called association neurons. And those are between the motor and sensory neurons. So they serve in a way to take information back and forth from motor to sensory and vice versa. Okay. Most of those are inside the CNS. All right. So we see here that most of the body's neurons are in interneurons. So here, just looking again at the structure of the neuron, be very familiar with what this looks like, right? That you have these processes here, there are the dendrites. So here you're going to have all types of information. They're going to come through these dendrites, and we are going to look at another type of potential, but that is not the action potential that we have seen before, right? And how that is going to influence the information that will go down the axon or not, okay? Remember that this initial part of the axon, leaving the cell body, you have a very important region that is called the axon hillock. And today we're going to look into more details mechanistically how that is working and how you get that propagation of the impulse that either goes or it doesn't. Okay, so here's a pretty picture. What type of neuron is this? Multipolar, is that it? Everybody got multipolar? Sure. Yeah, kind of undecided. How many people think he's multipolar? Hands in the air, up. It's time for you to vote, yes? Good. How many people think it's something else? All right, I'm not gonna ask who's not thinking, okay? So here, soma is the same thing as the body. Dendrites, what gets the stuff to the soma, and there you have your axon, and there you have your synapse, connection to something else. All right, here is another question. Each neuron in your bodies has a lifespan of, you should have seen that with your homework. And you should also think about what happens in case you damage a neuron. In case you have, for example, a spinal cord injury. If you damage a neuron badly enough, yes, most people got it right. Each neuron in your body has a lifespan that is about your whole life. Okay, it will stay there as long as you don't damage it badly enough for your whole life. Okay. All right, so we have mentioned, I believe I had mentioned this before, or, oh, I like the ringtone. Okay, so you have to know the differences of what is happening in your central nervous system myelinated axons in comparison to your peripheral nervous system myelinated axons. You have to know which type of cells is responsible for myelinating the axons, right? Because they're different cells, they work in different ways. But also you have to know the principle of what is happening to the action potential propagation that is going to go on that axon, right? That is myelinated. Okay. So here, the myelin sheets in the central nervous system. So those are going to originate not with the whole body of the cell that we have seen before, right? This is not the whole body of the cell. This is going to be only processes or things that come out of the cell body, those little appendages, okay? So those are processes of this funny looking cell that is called oligodendrocyte. Now the cell name is telling you what this cell has. Oligo for a lot, dendro for dendrites. So you can imagine these things are coming out of the cell body, okay? Many dendrocytes, many dendritic arms, or many processes, sites is for cell, okay? So oligodendrocytes are those cells that are going to stretch out a lot of arms and cover the axons of a lot of neurons. Sometimes one cell can wrap up to 60 axons at once, okay? So this is a very interesting cell body with 60 arms wrapping up different axons or different parts of the axon, the same axon. Okay, so remember, this is what this thing is looking like. And this is all part of your central nervous system. What is the name of the cell that does the same type of job in the peripheral nervous system? Schwann cell. Everybody got that? What is the cell? Schwann cell. All right. And this guy is oligodendrocyte. Okay. Now, okay, let me see here. So when you look in the central nervous system, right, and you see something that looks whitish, okay, hopefully we think most of the time it's not, well, it is kind of fat, but in this case it's not just fat, it's a specialized type of fatty tissue, okay? So if you look at something and you see white matter, that is in something that is myelinated, okay? So myelinated fibers, those are, they show up like white matter, okay? So if this is myelinated fiber, what is a gray matter? It's not myelinated, it's something else out of this neuron. So the gray matter is basically going to be like the cell bodies, the part of the neurons that do not get myelinated. Okay, so gray matter, cell bodies, and then white matter is myelinated axons. Okay. All right, so now we get into a lot of details on membrane potentials. So if you didn't get it the first time when we were looking at the muscular contraction, muscular function, neuromuscular junction, so now for sure you're going to get it about the action potential. Okay, you're going to finish this class knowing everything you can possibly know about the action potential. You're going to be dreaming with action potential channels and what happens in each type of voltage and what the voltage means. What is the voltage anyway in the first place, right? So make sure you understand that, because now we're really going to go into a lot of detail how this thing is working. Okay, so we know about the resting potential. We're going to talk more about that now. Okay, and basically we know that neurons, just like muscle cells, those are excitable, right? And we did have a lot of that in the neuromuscular junction. So make sure that you refresh your memory as we go through this more detailed view of the action potential. Okay, so we have seen that action potential here, and the transmission of signal is going to happen at the synapse. So here you have your presynaptic terminal, and then you have your postsynaptic cell. And then the thing in the middle is the synaptic cleft. Okay, so let's see what is happening here. You can have several types of potentials. They're not quite the action potential. Okay, those are summation type of potentials. So what happens is that if you have some type of depolarization, it means that the cell is going to be more positive than its resting potential. So keep that in mind. Depolarization is really getting the cell membrane more positive than it was at rest. When you talk about hyperpolarization, is really getting the voltage at the membrane to be more negative than it was at the beginning, okay, at the resting potential. Now, one principle or one association that you have to get from this, depolarization signals are more apt, or they are more conducive of generating an action potential. Okay, the more depolarization signals you have, the more the chances are that you're going to get an action potential out of this. All right. In contrast with the hyperpolarization, they tend to be negative signals. It makes it more difficult for the neuron to generate an action potential. Right? So when you have hyperpolarization, it takes a lot more effort from other signals that can be depolarizing. It takes a lot more of those signals to get this thing to fire an action potential. So keep that in mind. From outside, comparing the outside to the inside of the cell, this charge here is going to be reversed if you only have sodium channels open. So this here, instead of minus 70, it would be positive 60. That's odd, right? That's not something that we are familiar with the resting potential. Okay. Now, that would be the case if you would only have sodium channels open. So inside of the cell gas positive is if all the sodium channels break loose, right? Now, if you close all the sodium channels and you only open potassium channels, that ion is going to behave differently. It will tend to just leave from the inside of the cell, right? And when it leaves the inside of the cell, oh, I forgot to mention that the potassium, the higher concentration of potassium is usually of sodium is outside of the cell because of the sodium-potassium pump, right? So it will tend to enter the cell. That's why the inside of the cell gets more positive if you open all of the sodium channels, right? Now, coming back here, if you open all of the potassium channels, so since the inside of your cell has more potassium than the outside, this potassium will tend to get out. Now, when something positive gets out of the cell, the inside of the cell becomes more negative, right? So with the inside of the cell becoming more negative, if all of those are open, if you keep them open, then and the sodium-potassium pump working, then your cell would be at a much lower resting potential than it actually is. Okay, so keep that in mind, that the equilibrium of chemical and electrical properties of these ions, that those are different. Okay, is everybody okay with that notion? All right. Okay, so let's see, we're going to look into details what is happening at each stage of this action potential process, at each stage of channel response based on the environment and the stimuli that are getting to the area right around the channel. Okay, so here, when we look at first at the resting potential, right? So we know that resting potential was reached because of sodium potassium pump, right? So at the resting potential, you have this negative overall energy inside of the cell. And your sodium channels are closed. They're in the closed position. They're not blocked. They're not inactivated. They're just closed, right? And then your potassium channel is also closed. So you have a lot of potassium here inside the cell. You have a lot of sodium out there of the cell, right? So now, let's say that you have a lot of events that are coming to this neuron through the dendrites, right? And overall, they are depolarizing. So you keep on having depolarizing events that, remember, depolarizing means that the inside of the cell gets to be less negative or more positive, right? Now, if you get enough of these events that you allow this to reach negative 50, how far is this from minus 70? What is the jump here? 20, right? 20 millivolts. So if you put enough charge of 20 millivolts to allow this overall area of the membrane to reach minus 50, something is going to happen. This channel that is a voltage sensitive channel, remember we have seen a similar channel before that was ligand gated, but this guy is not ligand gated. You don't see a ligand stuck up there. You see this guy is actually a voltage sensitive channel. Okay, it will feel the difference in the voltage around itself, right? So when it reaches, when the cell around here reaches negative 50, this channel is going to open very, very quickly. Now, somebody had asked me before, why is it that the potassium channel did not open, right? And I say, well, because it is slow. Not just like it is slow, it doesn't like to open at that voltage. It has to have a much more positive environment of charge, of voltage. It has to have a much more positive depolarization for that thing to open. They have different sensitivities depending on the voltage that is in their environment. Okay, so at minus 50, sodium channels are going to open, and they're really, really fast at opening. Okay, so once the sodium channels open, what is going to happen to sodium? Sodium is going to enter the cell. What is going to happen to your membrane potential? It's going to go up, right? So, okay, so that's going to go up. Now, this here was from also one of the exercises that you have in your homework, right? So what happens here is that once this membrane potential is reached, that is more positive, right? So you have here some positive charges that went here. So this is overall minus 50 millivolts, right? So once you have one of this channel opening, now the inside is going to be a lot more positive, and that's going to trigger a positive feedback with all of the other sodium channels that are around this one. Okay? You got the picture of what's happening here? All right, so positive feedback once you reach that threshold. And this is what it said, that once you reach that threshold and this sodium channel open, that is the point of no return. That means your action potential has to be fired. Okay? So we're going to go into more details of how that is happening, right? So this, when the action, when the membrane potential goes a lot more positive, that means sodium channels are open, because sodium is really going to make it more positive. Okay, so here is the action of sodium channel opening. Now those sodium channels are, once they are open, you know potassium channel is closed, because they are waiting for a very positive environment to open. Like negative 50, that's not good enough for them. It's still negative. They want something positive, so they will open with something positive, okay? So now let's see what's happening here. With these sodium channels, as we have seen before, it flushes a lot of sodium inside the cell, and then it gets inactivated, right? So at the time the channel gets inactivated, that's the time that the inside of the cell stops becoming more positive, okay? What is the voltage that gets the sodium channel inactivated? Yeah, I know you got it. Okay, let me see somebody from the back. Any guess? This is the audience participation part. Okay, the question, like, what did I ask? At what voltage is sodium channel inactivated? Look at the chart. This is time, this is voltage, increasing voltage going like that. Huh? Positive 30, yes, everybody sees that here? Look at this. Look at the graph is telling you. At positive 30, right there, something is happening here. You don't get this action potential that keeps on going and going and going, okay, up. No, that doesn't happen, right? At positive 30, that channel is going to feel that the environment is too positive for it, and it doesn't want to be open anymore. So it's going to put that ball and plug itself, okay? So that guy likes negative 50 until positive 30, and then it shuts down. It gets inactivated. At positive 30, that's the environment, the positive environment that the potassium channel needs to get itself open. Remember, it doesn't like to be open when it's negative. But when the membrane potential reaches positive 30, potassium channel is going to open. Once the potassium channel opens, what is going to happen with potassium? It's going to leave the cell. So something positive is going to get out of the cell. What is going to happen to your membrane potential? It's going to start to go down. Okay? So that when potassium is going out, that is the part of going down. Going down. All right? That's the part that your membrane potential starts to get repolarized. So what is responsible for the repolarization of your membrane potential? Potassium channels. What type of channel? Voltage gated channel, not leakage. Voltage gated potassium channels. Okay. All right, so now you have your potassium channels open here. Your membrane repolarization is taking place. Now, until this whole membrane potential reaches back to negative 60 or so, until that happens, your sodium channel is still inactivated, but it comes to a point that the membrane is going to be negative enough that is going to allow that inhibition to get out and the potassium channel to close. So the sodium channel to close. So no more sodium is going to be getting into the cell. But at the same time, at that part, once you reach that negative value, your sodium channel could be excited again if you have a big enough stimulus. Now, this inactivation of your sodium channel during this peak of your action potential, that is what prevents this action potential from going backwards. It's type of a safety mechanism. So the direction of the potential is always going to be unidirectional. Okay, that's an important concept that sometimes is not really understood well. Everybody got that? Okay, so why is it that the thing doesn't fire backwards? Because the sodium channels get to be inactivated. Okay, all right. And then once the membrane is repolarizing, after this stage, then the sodium channel is going to be closed, but it could be activated again. And at this stage here, you also get your potassium channels to close, like right here, okay, at the bottom, at the hyperpolarization stage. And then your sodium-potassium pump is going to reestablish your original membrane resting potential. Okay, now, the magnitude of the action potential, like how big that voltage change is, intuitively speaking, is from the resting potential where it started to the maximum voltage that it reached, right? So that's the difference. So what is your magnitude of action potential here? Sorry? 100 millivolts, right? Okay. All right, so here is just a bit more of an explanation of what that activation and inactivation looks like. Keep in mind what is happening at each of those voltages. Right, minus 70 is closed. Minus 50, that's the threshold when it opens. Positive 30, that's the threshold, that's the voltage, not the threshold. Threshold is for when it's open. That's the voltage that is going to inhibit the channel, to plug it, okay? And it's going to stay inhibited until this thing goes back to resting potential voltage. Okay? So here is the behavior of your potassium channel. It is closed at resting. Is only going to open when you reach positive 30, right? Positive 30, so it's going to stay open until you go all the way past your resting potential, all the way down to negative 80 millivolts, hyperpolarization. Then it will close and it's going to remain closed at resting potential. The action potential is an all or none type of response, because once your sodium channels open, they cannot just change their mind and close right away, because they depend on the change of the voltage to close again. They are only going to inactivate when this voltage reaches positive 30 millivolt. Okay? So it's not like they open it like, oh, I changed my mind. It's like, forget it. It cannot change his mind. Right? It has to remain open until that reaches positive 30 millivolt. Okay. Now, how fast is the action potential going down the axon? Okay, so that has to do with three basically parameters. First thing is how thick the axon is. If you have a very thick axon, that means a lot of action potential can go down very fast. Okay, so if you have a thinner one, that means your action potential is slower. Okay. Also, degree of myelination, because remember, when you talk about myelinated axons, you have those things that are the saltatory action potentials, right? And they're happening inside of the axon, and they go very, very fast because they don't have to depend on channels opening and closing during that insulating, across that insulated part of the neuron, okay? So the action potential, it jumps, like it's a little trick that the ions inside of the cell, they just bump into each other very fast to reach the next area where you don't have the myelinated insulation, and those are your nodes of Ranvier. So there, at the nodes of Ranvier, you're going to have sodium and potassium channels. They're going to behave the same way as we have seen so far, depending on the voltage, on the potential of that across that membrane. Now, so these are the two types of conduction that we have: continuous without myelin, saltatory with the myelin sheets. Okay. All right. And also how, of course, how long the axon is. The longer the axon, you know, the longer it's going to take for the action potential to get down there. That's like quite intuitive. So three things that are going to interfere, or they're going to contribute to velocity of the action potential. One is the diameter of the axon. Two is whether it is myelinated, and the third thing is how long that axon is. Okay, so here is also straight from your homework, one of the activities in your homework, comparing how fast that action potential goes down the axon. Okay, larger diameters have less internal resistance. Another thing that you can visualize is that when you have a large diameter, you have a lot more probability that ions are going to bump into each other here, because you have a lot more ions that are present here in comparison to that amount. You can put that in terms of how quickly that propagation can take place. But then when you compare with a myelinated axon, this one wins the whole thing. I mean, it is even thinner than the other one here, but because of the saltatory action potential, then the whole electrical signal can go down a lot faster. Okay. Another homeostatic imbalance, that is on multiple sclerosis. So in multiple sclerosis, you have antibodies that your body is producing, that they attack the myelin sheets in your central nervous system mainly. Okay, so once the myelin sheets are attacked, then they become some type of scar tissue or hardening tissue that is called sclerosis. Okay, so multiple sclerosis is really going to interfere with how your nerve impulse is, or how the action potential gets transmitted. And it is a progressive neurodegenerative disease, and basically there is no cure, and it's only palliative type of care. Okay, and it leads to a weakness, muscle weakness, visual disturbance, loss of muscular control, loss of speech, and incontinence. You know what is incontinence? can hold urine or feces. That's incontinence. Kind of sad. All right. Now, there are some drugs that can inhibit the immune system attack on those myelin sheaths. So those are immunomodulatory drugs. Okay. Interestingly, of course, this cannot be prevented since it is an autoimmune disease, but interestingly, proper level of vitamin D can reduce the developmental risk of the disease. Where do you get vitamin D? Out in the sun, or drink milk, right? Okay, out in the sun. Now, how would vitamin D avoid development of multiple sclerosis? Actually, vitamin D is very important for proper immune system function. Okay, it's not like going to activate the immune system. Otherwise, you would be activating the cells that are attacking your neurons. So that's counterproductive. What it does is that it modulates, it makes the function of the immune system better. So the immune system is not going to produce the antibodies to attack the body. Instead, it's going to behave in the way that it's supposed to behave in the first place. So vitamin D is very, very important for proper work of your immune system.